Okay, today I'm going to talk to you about belt transects. So I'm going to write underneath the purpose of belt transects. It's to find out how organisms are distributed across a gradient, and explain what that means in just a second. Across a gradient. So, first of all, an organism is a living thing. So we might be interested to see how living things change across this thing called a gradient. And what a gradient is essentially a change in an abiotic factor. So an abiotic factor is a non-living factor. So we might be interested to see how, let's say, light intensity affects the growth of plants. We might be interested in looking at how the amount of water in the soil affects the growth of plants, or the temperature. Basically, how a non-living thing affects the distribution of a particular organism, a particular living thing. So I'm just going to imagine here, I'm just drawing a very simple diagram of a river. So imagine we have this river flowing here. Now what we might want to do is we might want to see how does basically the further we get away from the river, how does this affect the distribution of organisms? So the abiotic factor in this particular case that we're looking at would be the level of water that we find in the soil. So the first thing that we do is we get a tape measure and we set it up so it goes away from our particular starting point of interest. This could be a river, it could be the end of a line of trees in a forest, whatever we want. So what we need to do is this. We use a bit of equipment called a quadrat. Now all a quadrat is it's a square frame that allows us we put it down on the ground, and it allows us to count the organisms of interest within the different gaps. So we'll be counting to see how many there are. So what we need to do is essentially we're interested to see how does the level of water in the soil affect the number of plants. Let's say we're looking at daisies, the further we get away from the river. So what we do is we have a starting point, which would be right here, where we put our quadrat down. What we then want to do is we want to reposition our quadrat at equal intervals, slowly going away from our particular point of interest. So let's say, for example, if that one's at 0 meters, this one's at 2 meters, then 4 meters, then 6 meters, then 8 meters. So every single time, what we're going to do is when we put down the quadrat, we're looking at two things. First of all, we need to count the number of organisms that are within that quadrat. And secondly, we need to use some kind of probe or sensor in order to figure out the kind of how much of that abiotic factor we have. So in this particular instance, what we're going to be using is a water sensor to see how much water is in the soil here, 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 and here, and counting how many plants or how many daisies we have in each of these different quadrats as well. Now, different abiotic factors we could investigate. We could be looking at the effect of light intensity. So we might need a light meter at each of these settings to read how bright it is. We could also use a temperature probe to see the temperatures at each of these places. So after we've done the belt transect once, we just need to repeat it a couple of times in order to collect more data. So again, exactly the same technique, but this time a little bit further along. So here's my 5 quadrats that I put down again. And then I need to repeat it at least 3 times. And each time I'm putting down the quadrat, I'm counting the number of organisms, or in this case daisies, that I'd have at each of these intervals. Again, evenly spread, evenly distanced. So once I've done that, what I can do is I find my average number. I take the mean number of plants for these 3. So I add up the number of organisms found in each of these 3. And in this case, because there's 3 of them, I divide by 3. I'd do the same for the abiotic factor. So I'd find the reading on my water sensor, and I'd find the average, so the mean value for these 3. Once I've collected all the data, I can then hopefully draw a conclusion to see how does the level of water affect the distribution of daisies along a belt transect. And that's basically it. Thank you.